Alright, welcome back, guys. This is Nurse Franny. This is episode 4 on Let's Study Anatomy and Physiology. Today we're going to go over genetics. We're also going to go over histology, a brief overview of the integumentary systems and the skeletal system. We're going to briefly go over them as a little study guide on how to create nice flashcards. So the first thing that you want to be able to master is the law of complementary base pairing. Okay, and the definition, or what you want to keep in mind, is during a test is, let's say that the professor says that this base triplet, hold on, let me get a better stylus. So this base triplet, these three nucleotides we have, let's say thymine, we have adenine, we have guanine, right? Tag. So we have this base triplet that actually, I'm going to call it here, base triplet, it actually codes for one amino acid. Okay? So our messenger RNA, and I hate to say this, but it has like its base triplet of its own, but instead it's called a codon. The messenger RNA is going to go into the nucleus. Well, we find the DNA, we find all of these genetic instructions, and it's going to go ahead and write its corresponding or its complementary nucleotide. So we have adenine. When messenger RNA sees thymine, it calls for adenine. When it sees adenine, though, however, it actually transcribes uracil. All right, that's the only difference between RNA and DNA at the moment. That we're replacing thymine for uracil. And when we're coding for guanine, the messenger RNA actually a cytosine. So same thing there. Now, messenger RNA went ahead into the nucleus right here where we found the DNA. It transcribed, all right, I'm going to write it right here, transcription. It went ahead and transcribed the information onto itself. The next step, or step number 2, messenger RNA will go ahead and call out for transfer RNA. And transfer RNA is going to do the same thing. It sees adenine, law of complementary base pairing says it calls out for uracil. It sees uracil, and the law of complementary base pairing says that it needs adenine. It sees cytosine, law of complementary base pairing, guanine. These three are called the anticodon or anticodon. I hope I'm pronouncing it right. Okay? And I'm going to do a little visual on what this looks like, or what I think it would look like physically in a cell. So right now we have, for your flashcards, we have your law of complementary base pairing. You need to know what a base triplet is. You need to know what a codon is, an anticodon. It'll be good to know what DNA polymerase is. Polymerase, which corrects, mistakes when it comes to the transcription process. We also need to know what messenger RNA is and its function, and transfer RNA and its function, which we already saw. We know that messenger RNA does the transcription process, and transfer RNA does the translation process, which is the actual synthesis of that amino acid. Now we have those. We saw kind of in theory what that is, and now we're going to go and try to actually draw it. We're going to try to draw it. Discard that. Okay, so let's say I have a cell, and this cell right here, I'm going to draw it with its phospholipid bilayer right there. So this is the plasma membrane of a cell. Now we're going to draw the nucleus And it also has a double membrane. And now I'm going to zoom in. This is a nucleus. Now I want to draw a little, hold on, little nucleolus. Oops, sorry. Let me move that if I can. Jesus Christ. There you go. Now this is the nucleolus. Now, DNA, you can actually find it in here. So you can find your DNA in here. Okay. And here we have our endoplasmic reticulum. And here I'm going to draw some little circles if I can. These are ribosomes, and these are adhering to the endoplasmic reticulum, and this portion right here we're going to call the rough ER. Sorry. Jesus. ER. And this little portion right here we're going to call the smooth ER. But around we also have some free-floating ribosomes. We have one here. We have one here. Oops. We have some over here, some free-floating ribosomes. Okay, now step number 1, we have our DNA right here. However, messenger RNA wants to go in here and transcribe. Transcription. It wants to use transcription to actually copy the information found in here onto itself. Once it's in here, then it's going to leave the nucleus and bind to a ribosome. It's going to bind to a ribosome. Once it does that, it's going to call out for transfer RNA. Transfer RNA is going to come here, boop. And then these guys are going to do what is called translation. Okay. So this process, once that happens, right? Step 1, messenger RNA. So I'm doing different colors. Step 1, messenger RNA goes into the nucleus. I feel like I'm Madden right now. Goes into the nucleus. And transcription occurs. The next step, it leaves the nucleus, binds to a ribosome, and translation occurs. Then your amino acid is synthesized. And you need a lot of amino acids. You need a lot of little amino acids to actually form what we know as a protein. So this is going to happen hundreds of thousands of times. And this we're going to call here, this little guy right here, is finally going to be our protein. Okay? Now, very important to know that there's a bond between every single amino acid. And that bond is called a peptide bond. And that's the next thing that you should know. So your peptide bond is another of your flashcards. So